Hello, dear students. Myself, Sunita. Welcome to this session. In this session, we shall be learning about the next type of cell division, that is meiosis. In our previous sessions, we have learned about the type of cell division, that is mitosis, and its phases, also about the significance of mitosis. So, in this session, meiosis is what we are going to understand. So, what exactly is meiosis? And what is meiosis otherwise called as? Where exactly meiosis will occur in case of plants and in case of animal cells? And also we'll be understanding the different phases which is involved in meiotic division. And lastly, we'll be understanding about the significance of meiosis. So let's go ahead and learn about meiosis in our session. So basically, meiosis is derived from a Greek word. Meom means to reduce, or osis means state. Meiosis is called as reductional division. It's called as reductional division. That is why it is called as a reduced state. So when we look into the number of chromosomes from the parental cell, this is a parental cell, which is going to divide itself into the daughter cells. So the chromosome number in case of parental cell, if it is 46, they're going to distribute to the daughter cells into half. It will be reduced into half. So this type of cell division where a diploid number of chromosomes, if this is a diploid number of chromosomes where the chromosome number is represented by 2n, that is the diploid condition, when it undergoes meiosis to form the daughter cells, and these daughter cells are reduced into half. So we call this, and the condition is haploid here. This type of division is called as meiosis. Since there is reduction in the number of chromosomes, so we call it as reductional division. So reductional division is important because to maintain a constancy of chromosome number in sexually reproducing organisms. So sexually reproducing organisms, basically they are multicellular organisms. So they need to maintain a constant chromosomal number. So parental generation, if it is diploid, then the daughter generation will be haploid. And it is most important type of division because it leads to the variations, and variation leads to evolution. So the daughter chromosomes, when the chromosome is passed on from one generation to another generation, that is from the parental generation to the offspring generation, the chromosomes, when they are divided among the daughter cells, the characteristic features of the parents are passed to the daughter cells. They may not be the same, right? In case of equational division or mitotic division, they resemble that of the parental generation. But in case of reductional division, the offsprings are not same as that of the parental generation because the chromosomes undergo a lot of variation. The chromosomes undergo recombination. So this recombination and the variation will lead to the diversity in the chromosomes. So that in turn will lead to evolution, right? So maintaining that is a very important step in case of meiosis. And it is only characteristic feature in case of sexually reproducing organisms. Then what are the steps which are involved in case of meiosis? Meiosis process involves the interphase. Okay, it is involving the process of interphase, where the interphase is G1 phase, S phase and G2 phase. And this will be followed by the next phase, that is the meiotic phase. Okay, so meiosis phase will be divided into karyokinesis, which is followed by cytokinesis. But one thing here we need to understand is meiosis is divided into two steps called as meiosis 1. It's called as meiosis 1. Meiosis 1 has karyokinesis 1 and cytokinesis 1. And later that will be followed by meiosis 2. That will be followed by meiosis 2. And in turn, it will be followed by karyokinesis and cytokinesis, which will be again represented by the Roman letter 2. Okay, the Roman letter 2. So henceforth, meiosis can be said as it is characterized by single duplication of chromosome. We know that the duplication of the chromosome or replication of the chromosome will take place in the S phase, which is also called as synthesis phase. So since the synthesis phase or the overall interphase, there is only one interphase. So in the cell cycle, there is only one interphase, and that interphase is further divided into G1 phase, S phase, and G2 phase. So same events, same process will continue even here, but there will be only single duplication. There is only one S phase after the nuclear divisions or the cytoplasmic divisions. Okay, so there is no, say for example, meiosis 1 will be followed by directly meiosis 2, but there will be no, okay, but there will be no another interface in between meiosis 1 and meiosis 2. That is what is meant by single duplication of chromosome, which take place only at the S phase only once in the parental cell. And that parental cell will be followed by two meiotic divisions, which includes karyokinesis and cytokinesis. So karyokinesis is division of the nucleus, which has four important phases, which includes prophase 1, metaphase 1, anaphase 1, and telophase 1, followed by cytokinesis 1. And then cytokinesis 1 is followed by again meiosis 2, which includes prophase 2, metaphase 2, anaphase 2, and telophase 2, followed by the cytokinesis 2. So this completes the cycle in case of a cell which undergoes meiotic division. So it was discovered in the year 1888 by Edward Strasburger, and later meiosis was coined by Farmer and Murray in the year 1905. So this is the history behind the meiosis. So where does it occur? Meiosis occurs in the diploid cells. Okay, so we know that in the diploid cells. And in case of animals, these diploid cells may be spermatocytes, that is in case of the sperms, or they may be oocytes in the ovum. So sperm and the ovum are the male and female gametes which are produced for the process of fertilization. In case of plants, they are present in the specialized structures called as sporangia. Sporangia are the sac-like structure, and these sac-like structures are made up of many cells, and each of these cells are diploid in nature. And the cells which undergo the meiotic division or meiosis is called as meiocytes. They are called as meiocytes. The two meiocytes include the microspore and the megaspore mother cells of the plants. So which we call it as microspore mother cell, megaspore mother cell. In short, we call it as MMC. So let us look into the key features. I've already told you about the key features of meiosis. It involves two sequential cycles of nuclear and cell division called as meiosis 1 and meiosis 2. but only a single cell cycle of DNA replication that is going to happen in S phase. That is going to happen in S phase. Meiosis 1 is initiated after the parental chromosome have replicated and produced identical sister chromatids at S phase, involving a pair of homologous chromosomes and recombination between them. So ultimately, at the end of meiosis 2, 4 daughter cells are going to be produced, which are all haploid in nature. So these are the key features of meiosis. Now let's go ahead and look into each and every phase. So this is prophase 1. Prophase 1, in case of meiosis 1, is a complex and longest phase, which is further divided into 5 important substages based on the chromosomal behavior. Okay, based on the chromosomal behavior, it is divided into 5 substages. You can see in case of the interphase, the nuclear envelope, the chromatin material, the centrosomes with centriole in case of animal cell, they have already duplicated and now they are ready to undergo the process of prophase 1. So prophase 1, for the convenience, is divided into 5 substages based on the behavior of the chromosome. So based on chromosomal behavior, they are into 5 substages, which includes leptotene, zygotene, pachytene, diplotene, and diakinesis. Here, in case of the structures, the chromosomes, again, in the first phase, they undergo condensation and later they may undergo the crossing over. Recombination takes place between the homologous chromosomes. A exchange of genetic material will take place, so which plays a very important role. Henceforth, prophase 1 is a complex process and it is the longest process. It takes a lot of time to undergo the process of nuclear division. Let us look into the events which take place in prophase 1. But before that, you can just have a glance of the leptotene, the first phase, then comes the zygotene, then comes the pachytene, and then comes the diplotene, and then diakinesis, and also which later undergoes meiosis. In meiosis 1, the metaphase stage 1. So first one is meiosis 1. In meiosis 1, the prophase 1. The first stage, the first substage is leptotene. This is the substage. The word leptos is meaning thin. Tinea is band, nema means thread. So that means thin thread-like structures are present in the form of chromosomes in case of leptotene. Here, just like in case of prophase of mitosis, the chromosomes are thin thread-like structures which undergo the condensation process. So we know that during the condensation, dehydration will take place and the chromosomes become thin thread-like structures. And these thin thread-like structures are visible under even the normal microscopes. And they undergo the process called as condensation followed by spiralization, which ultimately later goes to the next step. So chromosomes which have undergone duplication, the chromatids remain invisible, but the chromosome thread-like structures can be visible under microscope. This is an animal cell which is depicting the chromosomes, and each chromosome is held by the centromere. Okay, these are the centromeres which are holding the chromatin threads. And also we can see that the centrioles develop asters. This will be taking place only in case of animal cells. Okay, only in case of animal cells. So when we look into the chromosomes, they appear in the microscope just like beaded-like appearance. You can make out the beaded-like appearance just like this. The thin threads which are constituting the bead-like structures. And these bead-like structures on the chromosomes are called as chromomeres. When we look at the chromosomes, the chromomeres are the bead-like structures which are visible on the thread-like structures. Centromere is the center of the chromosome, whereas chromomere is the visible portion which looks like bead in appearance. So in case of animal cell, they form the bouquet stage, and that is how they appear when we look under the microscope. So this is the leptotene stage. So, and now later, when it enters the next stage, that is zygotine stage, the end of the chromosomes are attached to the nuclear envelope by a structure which is called as attachment plate. So these attachment plates are majorly made up of proteins. And now later, the further process will continue with the next substage that is called as zygotine. The word zygos means yolk, or it is called as pair. Tinea means band, nema means thread. So here the major event which is taking place is the pairing which happens between the homologous chromosomes. So homologous chromosomes are the type of chromosomes which are going to be formed from same type. Okay, so between the parental and the maternal, or you can say it as between the two homologous chromosomes, the pairing will occur. And that process of pairing is called as synapsis. Okay, so the pair which we are talking here is nothing but the synapsis formation. So during the synapsis formation, you can see here between the homologous chromosomes, which are depicted in two colors, blue and red. So the complex is going to be formed first, and this complex is called as synaptonemal complex. So that is nothing but the pairing which occurs between the two. They come closer to each other, and then they are going to pair themselves. So the synaptonemal complex which is formed will be like ladder-like appearance. Okay, and this ladder-like appearance will be due to the formation of the proteins which help in pairing process. So when synapsing occurs, the chromosomes do not lie side by side, but they rather present on top of each other with a protein called as synaptonemal complex which holds them together. So now the chromosome condition, the paired homologous chromosome condition is called as bivalent. It's called as bivalence. Bi means two. The valency is nothing but the referring to the chromosome pair. So these homologous chromosomes which are paired are called as bivalence. You can see how they are held together, the centromere along with the kinetochore, and how they are held together one top of the other and forming the synaptonemal complex. So during the synaptonemal complex form, they also form a structure which holds them even more tightly called as recombination nodule. Recombination nodule. Okay, they form recombination nodule. So this recombination nodules are also involved during the process of synaptonemal complex. More prominently, the recombination nodules are going to be seen when there is crossing over, which takes place in the next stage, that is Pachytene. So the first two stages of prophase 1 are relatively short-lived compared to the next stage, that is Pachytene. Pacus means thread. Here the paired chromosomes, that is the bivalents, they become thick and short. So due to the condensation process in the first stage, they are thin. Now here they become thick and short. You can make out how they are appearing in the microscopic view. They become thick and short. Okay, they become thick and short. And meanwhile, there's some important aspect which is going on, or an important event which is going on here. During the pairing of homologous chromosome, there is going to be exchange of genetic material between the homologous chromosome. So the exchange of genetic material will take place here, and that process is called as crossing over, which is the most important event which take place. So due to the crossing over, the genetic material is going to exchange between the homologous chromosome, and that is going to result in the further variations which are brought about in the daughter nuclei. Okay, so in the meiocytes, when there is genetic exchange which is undergoing, the process is called as crossing over. So as I told you, during the process of crossing over, there will be a appearance of recombination modules at the site of crossing over between the non-sister chromatids of the homologous chromosome. Generally, the exchange of genetic material will take place between the non-sister chromatids, between the non-sister chromatids of the homologous chromosome. So the genetic recombination can take place anywhere. That is of different types. So it may be at the end of the chromosome, or it may be somewhere in the middle. So that depends. And there is breakage of the enzyme which is called as endonuclease, and there will be reunion of this when there is breakage. Say for example, here there is the genetic crossover is taking place, so this is going to break and there will be exchange. When they are going to break into segments, later they are going to be forming the segments, and that will be done by an enzyme called as endonuclease. And later there is one more enzyme called as ligase which will join them. Okay, so two threads if you imagine, and then they are separated, and now these threads will be separated with the help of endonuclease enzyme, and later they will be joined by ligase enzyme. So henceforth, these two which are involved in the genetic exchange or genetic cutting and rejoining, they are called as molecular scissors and molecular glue in the molecular biology. Okay, these are the terms which are generally used for these enzymes. So now, when there is genetic crossing over, the genes or the chromosomal segments have also been undergone a lot of changes here, and that crossing over plays a very important role in the recombination of the genes in the chromosomes. So the bivalents here are forming the tetrads, and these tetrads are nothing but the bivalents which are formed of 4 chromatids. So you can make out these are the tetrads which are formed from these two. Okay, so bivalence is nothing but the chromosome. So homologous chromosomes, when they undergo pairing, they form the bivalents. Now these bivalents are composed of 4 chromatids, which are in turn going to form tetrads. So you can see the tetrads. Tetra means 4, so which have been represented in the microscopic view. So this is how the genetic exchange will take place between the homologous chromosomes. In a tetrad, how the crossing over can take place. So the crossing over can take place in any of the means, and you can see how the genetic exchange has been taking place. So during prophase 1 of meiosis, a part of the chromatid of one chromosome goes over to chromatid of the other chromosome, and vice versa. There will be interchange of the chromatids of a pair. This process is called as crossing over. So crossing over is a process by which 2 chromosomes of a homologous pair exchange equal segments. That is most important. So whenever there is exchange of genetic material, it is going to exchange equal segments. Okay, so crossing over occurs in the first meiotic division. Then next phase after the crossing over takes place is the diplotin stage, which stands the 4th substage. In case of the diplotin stage, diplos means double. Here the synaptonymal complex will dissolve. So which has been formed during zygotin will dissolve. And this during the formation, it forms an X-shaped structure. Okay, it forms an X-shaped structure. This X-shaped structure, which is formed during the crossing over, later, which will be dissolved, which is called as chiasmata. Okay, so chiasmata is the X-shaped formation which occurs during the crossing over, and now slowly there will be repulsion between the 2 segments, and the 2 segments of the chromosomes, homologous chromosomes, because the synaptonymal complex is going to dissolve. So you can see here how the genetic exchange has taken place here. Okay, and how the crossing over has been lit. Okay, even here, and chiasmata formation will be occurring. Then, apart from this, this is the most important stage, especially for women. In case of human female, the diplotin stage will be prolonged and it will be arrested for about 8 to 10 years. Okay, it will be arrested in the same stage for 8 to 10 years in human female, from 5th month of fetal development up to 12 year during the start of ovulation, that is when she attains the puberty in the oocytes of the vertebrates, especially in case of the human and also in case of some mammals, it will occur. So this stage is most important because in case of female, it will be an arrested stage, and later when she attains the puberty, it is going to she is going to once again attain the puberty, and then the ovulation will occur. So these are the important events which take place during the diplotin stage. Coming to the last stage, that is diakinesis. Diakinesis, dia means across, kinesis means movement. So during this stage, now if you remember the telophase, the condensation, once again there will be uncoiling, and that is how the chromosomes were now it became thick. Even here the chromosomes become thick and short and condensed, especially when it is attaining from, in case of mitosis, it was from prophase to metaphase. Now same thing is going to happen here. The chromosomes become thick and short and condensed, and the egg-shaped structure which was formed, and now slowly, due to the synaptonymal complex which has dissolved, the egg-shaped structure, now you can see here how the egg-shaped structure was formed, and once again it is going to repel and they are going to separate themselves. And that process of egg shape which has end is called as terminalization. So terminalization is a process where the chiasmata slide towards the tip. They disappear one after the other by moving towards the tip of the chromatids, and that process is called as terminalization. So terminalization, along with terminalization, even the nuclear membrane and nucleolus will disappear. The spindle fibers begin to form because they are going to attain the next stage, or they are going to enter the next stage, that is called as metaphase. So there is a transition again between the prophase 1 to metaphase 1. Okay, so now the chromosomes, they start, especially the tetrads, they are going to start getting attached to the spindle fibers through the kinetochores, and now get ready for the next stage, that is metaphase 1. So this transition stage is also called as prometaphase. It's also called as prometaphase. So these are the 5 substages of prophase 1. You can remember by the formula known as LZP D square. So leptotene, zygotine, pachytene, diplotin, and diakinesis. So these are the substages on the whole. You can just go through this. What are the major events in each and every place plays a very important role for you to answer some of the questions. Let's go to the next phase, that is the metaphase, later anaphase 1, telophase 1, and cytokinesis. So you can see the overall picture. So now the prophase 1, this is how the bivalents or the tetrads have been now undergone the process of recombination and getting ready to align themselves on the equatorial plane. Now they are going to separate themselves, move towards the pole, and then divide into daughter cells. So that these are the processes which are going to take place next. In metaphase 1 is the second phase of meiosis. Here the tetrads from prophase 1, they are going to line up in the middle of the dividing cell randomly. And so initially they have got ready, and now they have been aligning themselves at the middle. So again, on the equatorial plane, or it is also called as metaphase plate, the spindle fibers, they attach themselves the spindle formation would have already started in animal cell from the centrioles. They all start forming and then they are going to attach to the kinetochores, okay, on from either of the poles. So again, they may be continuous, discontinuous, or they may not be reaching the poles, or they may be reaching to one of the poles. So that depends on the type of spindle fibers which are present. So in case of plant cells, there is no aster formation. In case of animal cells, there is going to be aster formation. So the same conditions are applying here. It's called as astral meiosis in case of astral meiosis in case of animal cell, and anastral in case of plant cell. And once they attach themselves, they are going to align themselves on the equatorial plane, which plays a very important role again for the distribution of the daughter cells. So here in case of metaphase 1, there will be two metaphase plates which are going to be formed. You can see here, this is one plate and this is one plate. So two metaphase plates which are parallel to one another are formed. And each set of chromosome is connected to only one pole of spindle fibers. So each set of chromosomes you can make out here, it is either set to one or either set to the other. And both of the kinetochores of homologous chromosome, they become connected to the same pole. Okay, that is something special regarding the metaphase here. Even though the spindle fibers are formed, but the other spindle fibers which are not involved, but major spindle fibers which are involved here is only the one which are going to be attaching themselves to the either of the poles. Okay, so these are the major events which take place in metaphase 1. Now going ahead with anaphase 1. So you can see here, so how the spindle fibers are attaching themselves to the kinetochore. The homologous pairs of chromosomes are held together and how they get attached themselves. During the anaphase stage, the X-shaped structure, that is chiasmata, will generally disappear, and the homologous chromosomes, they are going to get separated and move towards the opposite poles. So ana means up. The phase is the stage where they are going to move towards the opposite poles. So especially the spindle fibers, they are going to pull again, and that process of pulling is called as disjunction. Okay, this process of pulling is called as disjunction. It's called as disjunction. So here the centromeres do not divide. They are tightly holding the chromosomes. The homologous pairs are tightly held by the centromeres, but they do not divide. And the sister chromatids, they remain together. Okay. Now the chromosome itself, they are going to separate, and they are called as univalents, or they are called as dyads. So these are the chromosomes which are called as univalents or dyads. So you can see here how they are moved towards the opposite poles with the help of the spindle fibers, and they are called as univalents, or they are also called as dyads. So because each of them, they have two chromatids which is attached to a single chromosome. So anaphase stage, the chromosomes are double-stranded. Okay, in anaphase stage, the chromosomes are double-stranded. And the disjunction which is caused at anaphase 1 is because of the independent assortment of the parental, that is the maternal and the paternal chromosomes, right? So this is a stage where they are moved towards the opposite pole. The sister chromatids remain attached at their centromeres and move towards the opposite poles. So this is the major event which take place during anaphase 1. Next comes the telophase 1. This is the last stage of meiosis 1. So in telophase 1, the homologous chromosomes pair reach the poles of the cell. Okay, they have reached the poles of the cell just like the mitotic phases. But the only thing here is the spindle formation will be uncoiling and they become elongated, and they do not reach the next stage, that is interphase stage, but they are going to undergo the next step, that is cytokinesis. So here the nuclear membrane is formed again, and there is decondensation of the chromosomes. The nucleoli membrane will reappear. Okay, the nuclear envelope reappear, and the spindles, they will disappear. If asters are present, in case of animal cells, even asters are going to be disappearing. Okay, even asters will disappear. And apart from this, now when we talk about the telophase 1, which is followed by cytokinesis, they are going to produce 2 daughter cells. Okay, so same thing will be followed. If it is an animal cell, it is going to form an cleavage furrow to form the 2 cells. These 2 daughter cells are called as dyads. These 2 daughter cells are called as dyads. So each daughter cells will be having only one pair of homologous chromosome. So it will be from either maternal or it may be from the paternal. So they will be haploid in nature. The chromosomes are said to be haploid in conditions, but the content of the DNA, since the replication process has already occurred in case of interphase, it is going to be double. If it is 2C, then it is going to be 4C, or if it is 1C, it is going to be 2C. So it depends on the type of the DNA content, how much it is present. Next is meiosis 2. So the phases of meiosis 2 includes prophase 2, metaphase 2, anaphase 2, and telophase 2. Earlier, in case of meiosis 1 was also called as reductional division. In particularly, meiosis is called as overall reductional division because there is reduction in the number of chromosomes from the parental cell to the daughter progeny. But when we look into the separate phases, meiosis 1 and meiosis 2, meiosis 1 is called as reductional division, whereas meiosis 2 is also called as equational division. Equational division, or it is also called as homotypic, whereas meiosis 2 was called as heterotypic because we saw the substages in case of meiosis 1, prophase 1. Homo means same type. So here it is same type of the division which is taking place just like that of mitotic division into prophase 2, metaphase 2, anaphase 2, and telophase 2, followed by the cytokinesis. So at the end of meiosis 2, they are going to be producing the 4 haploid daughter cells. So when we look into the chart of meiosis 1 and meiosis 2, so if this is a parental cell which is diploid, this parental cell will undergo the first meiotic division, meiosis 1, to form the 2 daughter cells. Now these 2 daughter cells are further undergoing meiosis 2 to form 4 daughter cells, which are again haploid in nature. So if we look at the chromosomal number, if it is 46 in case of human, the daughter progeny will be 23 and 23. Even here it will be 23, 23, 23, and 23. So the ultimate product of the end of meiosis 2 or meiotic division will be the 4 daughter cells formation, whereas in case of mitosis, it was only the 2 daughter cells formation. So that is the major difference between mitosis and meiosis. You can see how what happens in the prophase, the metaphase, the anaphase, the telophase. Ultimately, it is going to form the 4 daughter cells. So in prophase 2, Meiosis 2 begins without any further replication because there is no interface in between the meiosis 1 and meiosis 2. So the major events which are going to take place under prophase 2 is, it is not so elaborated, just like prophase 1 of meiosis 1. So it's a short phase. So same thing is going to happen just like mitosis, where the nuclear envelope is going to break, the spindle apparatus or the spindle fibers are going to form. Then the chromosomes, the centrioles are going to duplicate. And also we can see that the centrioles separate to form the centromeres. The nuclear envelope will break down and the spindle apparatus are going to be formed, are the major events which take place in case of prophase 2. And apart from this, the chromatids, okay, the chromatids will be condensed. They become thick and they become short, okay. The chromatids, they become thick and short. Okay. So this is going to be the stages in prophase 2. Next one, in case of metaphase 2, the chromosomes become arranged on the metaphase plate, just like the metaphase of mitosis, or just like the metaphase of meiosis 1. The metaphase 2, where the chromosomes will be now arranging themselves on the metaphase plate, which is also called as equatorial plane. So the chromosomes, as usual, they are going to be held together, especially at the centromere region, with the help of spindle fibers. So centromeres are arranged in a line called as equatorial plane of invisible spindle apparatus. So generally, the spindle apparatus will be not so prominently visible in meiosis 2, but the chromosomes are going to be taking the help of spindle fibers to attach themselves, because they have to be pulled in the next phase, that is anaphase 2. So the kinetochores here of the sister chromatids, they get attached to the opposite poles of the spindle through the chromosomal fibers and chromosomes, which are brought together by the process, and that process is called as congregation, or it is also called as congregation. So what is congregation? Congregation means it is bringing together the chromosomal fibers and the chromosomes together. When they come together, they are called as congregation. Okay. And later, the chromosomes move and occupy the equatorial plane of the spindle. But one thing we need to notice in metaphase 2 is, unlike in metaphase 1, there is formation of single metaphase plate with centromeres on equator and arm facing the poles. But in case of metaphase 1, we observed that there were two metaphase plate, whereas here there is only one metaphase plate. Okay, there is only one metaphase plate, and that too, it is facing the arms of the poles. So nuclear membrane has completely dissolved, or nuclear membrane has disappeared in case of metaphase 2. So going ahead with the anaphase 2, so you can also see here pro-metaphase, how they were attached, and now they have got separated. The sister chromatids are pulled by the microtubules attached to the kinetochores. So there the homologous chromosomes were pulled on either side, but now in case of anaphase 2, the sister chromatids are getting apart. So remember that anaphase 1, the homologous chromosomes, they were pulled each other, but in case of anaphase 2, the sister chromatids are pulled apart by the microtubules which are attached to the kinetochores. So what are the major events which take place in anaphase 2? The centromeres here separate, and the sister chromatids now individually they move towards the opposite poles. So you can make out Even here, there are different shapes which have been observed based on the position of the centromere. So the sister chromatids move towards the opposite pole, which is a major event which takes place in anaphase 2. And along with this, the separated chromatids are now called chromosomes on their own right. So the chromosomal fibers, which are majorly made up of microtubules, they attach themselves to the kinetochore, they shorten, they pull the daughter chromosomes towards opposite poles. The arms of the chromosome will gradually follow the centromere. So at the poles, what do we observe is each are having haploid set of chromosomes. Okay, so haploid set of chromosomes. That is, the condition of the chromosomes will be haploid. Now, from the content of the DNA, if it was 2C, in case of the initial parental cell, it would have been 1C. This is the DNA content. Okay, so metaphase plays a very important role again for the equal division of daughter cells. So 4 daughter cells are going to be divided during the anaphase 2. Later, it enters the next and last phase of meiosis 2, that is telophase 2. So during the telophase 2, the nuclear envelope forms around each set of chromosomes. So once again, there will be formation of nuclear envelope along with the other organelles. The nucleolus, which had disappeared, will reappear, and the chromosomes lengthen and it becomes indistinct. Okay, so the chromosomes will not be seen properly, but the length of the chromosomes will be longer, and also you cannot make out the chromatids and other arrangement, but we can make out that the chromosomes are present over there. So it's called as decondensation because it becomes thin and long. Okay, it becomes thin and long. It's called as decondensation. So spindle fibers are going to disappear here, and the chromosomes, they form a network which is called as chromatin once again. So telophase 2 is followed by cytokinesis. So after karyokinesis, that is the 2 daughter cells, they undergo simultaneous cytokinesis by the cleavage in case of animal cell or by the phragmoplast formation in case of plant cell to form the 4 daughter cells. So you can see here 4 daughter cells, either by the cleavage formation or by the cell plate formation. So ultimately, it leads to the 4 daughter cells. So each of these daughter cells have half the number of chromosomes of the parental cell. Okay, so each will be having half the number of chromosomes, and daughter cells are not alike. We know that due to the genetic recombination or crossing over which has taken place during meiosis 1, the daughter cells are not as same as that of the parental cell. Each of them, they have their own differences due to the genetic recombination. So unlike the twins, all other organisms are going to be genetically different, and this process is called as genetic recombination. So now let us look into, yes, you can see here the diagrammatic view of how the diploid parental cell undergone meiosis 1 and meiosis 2 to produce haploid daughter cells, which has undergone the process of genetic recombination. The exchange of genes would have taken place in prophase 1, and that has led into the combination, different recombination in case of daughter progeny. Looking into the significance of meiosis, just like mitosis, maintenance of constancy, as I told you earlier, is one of the most important significance of meiosis. It is one of the most important aspect of meiosis. So the constant number of chromosome has to be maintained from the parental cell to the daughter progeny. And due to this constancy in the number, it may lead to genetic variations. Okay, it may lead to genetic variations. And meiosis is most essential because it takes place in all the multicellular organisms. Especially this is going to take place in the germ cells or gamete formation cells, gamete forming cells, which are called as meiocytes. Okay, so, and that is most prominent in case of sexually reproducing organisms. So why maintenance, or why, what is the importance of maintenance of constancy of chromosomal number if we have to see? So, and they are specific to species. So it reduces the diploid number of chromosomes in the gamete forming cells, as I told you, that is the germ cells, to haploid number in the gametes. So that is why when we talk about a sperm or an egg, it will be haploid always in nature. And these two together, they ultimately leads to the fusion called as the sperm cell and the egg cell. They fuse to form the diploid cell, which is called as zygote. Zygote is the first cell of embryogenesis, that is formation of embryo, and that is how the whole body of an organism will be developed. So henceforth, this is most important. Genetic variations, we know that it has brought about because of there is recombination which will be occurring during meiosis 1, and that will lead to the variations, and these variations may lead to the process of evolution of species. Maybe due to this variation, evolution of species will occur. So we know that parental and maternal chromosomes, they are sought independently. That is why the variations will occur. And new combinations will be formed during the daughter formation. Lastly, it is essential for the formation of gametes in sexually reproducing organism, again, which will undergo the process of fertilization. So fertilization and sexual reproduction are the two important processes which are associated with the meiotic division. What if this process is not happening properly? So then that leads to the variations, and that may lead to even syndromes. So sometimes they may lead to trisomy, sometimes they may lead to monosomy, or sometimes they may lead to aneuploidy, sometimes they may lead to polyploidy. That is, there will be a lot of alterations which are brought about in the chromosomal number. So chromosomal aberrations or chromosomal variations which are brought about will be leading to different syndromes in case of the plants as well as in case of animals. Let us solve some of the questions. The pair of homologous chromosome in zygote is, the pairing of homologous chromosome in zygote is called as what? We know that the pairing of homologous chromosome in zygote is called as, it's not called as crossing over. Crossing over is nothing but the genetic recombination. So the pairing in zygote is called as synapsis. Okay, it's also called as synapse formation. Synapse is nothing but in case of the neurons where they are going to transmit the electrical impulses from one to another neuron. Chiasmata is the X-shaped formation, and crossing over is nothing but genetic recombination, which takes place in the next stage, that is the Pachytene. Next question. The nucleus of the diploid organism. So the nucleus of the diploid organism is about 3 ng, that is 3 nanogram of DNA in G1 phase. It is at G1 phase. Which one of the following statement describes the state of the cell at the end of S phase? So from G1 to S phase, what will be the content of the DNA is what has been asked here. So the nucleus divides into 2 and each nucleus contains 3 ng of DNA. The nucleus does not divide. We know that nucleus will not divide, isn't it? So henceforth, the two options are ruled out. But the nucleus does not divide and it contains 3 ng of DNA. Nucleus does not divide and it contains 6 ng of DNA. We know that after S phase, the content of the DNA, if it is 1C, it will become 2C. So henceforth, it will be 6 nanograms of DNA. Option D is right one. Next question. If meiosis 1 products have 20 chromosomes and 20 pgDNA, then what will be the number of chromosomes and DNA amount in G1 phase of meiocyte? Meiocyte is nothing but the cells which are undergoing meiosis process. So in case of the option 40 chromosomes and 20 pg of DNA, 40 chromosomes and 40 pg of DNA, 40 chromosomes and 80 pg of DNA, 20 chromosomes and 40 pg of DNA. So which is the option here? So we know that in case of the chromosomal number, since the number of chromosomes are going to be doubled, right? And the content of the DNA, which is nothing but in terms of picograms, or you can even refer it to as Daltons, okay? And what happens to the content of the DNA? So already meiosis 1 products have 20 chromosomes and 20 picogram DNA. Then what will be the number of chromosomes and DNA amount in G1 phase of meiocyte? So the option here will be option A, because the number of chromosomes will be 40 and 20, the content of the DNA will be the same, because here, since the synthesis phase would have already had 20 picograms, so it remains the same, but the daughter's chromosomes would have, the parental chromosome would have 40 chromosomes, which have been divided into daughter cells as 20 and 20. So hence the option is A. The enzyme recombinase is required at which stage of meiosis? So we know that recombinase is an enzyme which helps in genetic recombination. So which phase is it involved, a recombination at Packetine stage? Henceforth, it is the option D. Next one, at which stage of meiosis does the genetic constitution of gametes is finally decided? So genetic constitution, at which phase it is decided? We know that at anaphase 2, there will be whether the disjunction has happened or not, whether the chromosomes have been separated or not, will be checked at anaphase 2. So henceforth, this plays a very important role in deciding the genetic constituents of gametes, whether it has been distributed properly among the daughter cells or whether the disjunction would have happened. If the disjunction would have not happened, then it leads to a non-disjunction, or it is also leading to the syndromes. Next question. Meiosis involves. So meiosis involves how many nuclear divisions and how many chromosomal divisions? That is the cytoplasmic divisions. So we know that there are 2 nuclear division, meiosis 1 and meiosis 2, and 1 chromosomal division, which is the interphase. So option is option C. Next question. Exchange of paternal and maternal chromosome material during cell division is in which stage? Synapsis, dyad formation, bivalent formation, crossing over. So we all know that the exchange of genetic material, that is between the parental and maternal chromosome, will take place during the crossing over. So henceforth it is option D. Next question. Repulsion of homologous chromosomes begins during. So the repulsion of homologous, we know that it goes on beginning during diplotin stage, will ultimately leads to the terminalization in the next stage. So the stages in proper sequence of prophase 1, as I have told you, that you have to follow a formula called as LZP D square. So you can mark it easily. So the option here is option B. Okay. Next question. Meiosis 2 reduces the chromosome number from diploid to haploid is the statement 1. And the statement 2 is homologous chromosomes separate from one another in anaphase 1. So which are the statements which are correct and which is incorrect is the question asked here. So Meiosis 2 reduces the chromosome number from diploid to haploid. We know that meiosis 1 itself would have already reduced the number from diploid to haploid. So it will be continued by meiosis 2 as well. So henceforth, the second option says homologous chromosomes separate from one another in anaphase 1. Yes. So only this statement is right. So henceforth, our option is option C. Next question. Meiosis involves, I think the question has been repeated to children. We'll go ahead with the next question. The similarity between mitosis and meiosis is, so what is the similarity between mitosis and meiosis? Both are preceded by replication of DNA. Both occur in all kinds of cells. Both includes the division of chromosomes. Statements A and C are correct. So the similarities between mitosis and meiosis is that both are preceded by replication. S phase is there. It occurs in all kinds of cells. Is it so? No. In some cells, somatic cells, mitosis will occur. In case of germ cells, meiosis will occur. Both include the division of chromosomes. Yes, it is followed by karyokinesis and the cytokinesis along with the interphase stage. So only the two options are right here. That is option A and option C. So henceforth, our option should be option D. Okay. So rest all options are wrong. So our option should be option D. Thank you, students.